Today we are going to discuss cell membrane. Okay. So everything concerned about cell membrane is a very important topic. So listen carefully. Any idea about cell membrane? Anyone? You read in class 12, HS class 2 stage, preparing NEET exam. Anyone? No idea? Bolo.
The outer covering of the cell.
Okay.
It is basically a bilayer, lipid bilayer.
Very good. Anyone can modify this? It is called plasma membrane. Okay? Very important to listen carefully. So today we are going to discuss this thing, cell membrane, fluid mosaic model of cell membrane, membrane protein, specialized membrane structure, cytoskeleton, transport mechanism. Though I am going to discuss going to details in about transport mechanism because you will be read in physiology lecture classes. Okay, I will give you one overall brief idea about transport mechanism. So what is plasma membrane? There is a membrane covering outside the cytoplasmic structure. Okay. Is a plasma membrane is a semi-permeable. Why it is called semi-permeable? It is not permeable 100%. It selectively permeates some substances. It is called semi-permeable, right? They surround the cytoplasm of every living cells, separating its interior from the outside environment. Its structure and function are described by Fluid Mosaic Model. You may ask in the exam about Fluid Mosaic Model of cell membrane. Everyone read carefully. You may find one short note or part of long question. Okay, what is Fluid Mosaic Model of plasma membrane? It is proved to be that membrane is a dynamic arrangement of components that allow it to regulate the movement of substances and interact with other cells. What is fluid mosaic? This is the overall structure of cell membrane. You can see here, this is the cell bilayer. Okay, one layer, another layer, this is outside, this is inside. Here we have separated this layer. You can see here we have head and tail. Okay, this is the head, and this is tail. Okay, this is also head. This is outside of cell, and this is inside. This head is hydrophilic or hydrophobic? Hydrophilic. The tail is hydrophobic. Hydrophobic means it hates water. Hydrophilic means it interacts with water. Right? So it, like this, it make a bilayer arrangement, and then we have embedding. These are proteins, integral proteins, subglycoid proteins, globular proteins. We have some cholesterol in the cell membrane. Here you can see glycoprotein. These are proteins. Some carbohydrate moieties are attached with protein. This is glycolipid. This is cholesterol. These are integral protein. Okay, we are going to discuss one by one. So everyone understand? Everyone understand what is this? How the cell membrane is made up? This is made up of two layers, bilayer. We have head, that is hydrophilic hydrophilic head outside and hydrophobic tail inside. So it is hydrophobic, that is why it does not allow water to move freely. Right? Next. So this is fluid mosaic model of cell membrane. Fluid mosaic model describes the plasma membrane as a flexible and dynamic structure. Membrane composed of phospholipid bilayer. Okay, we discussed already. Phospholipids are arranged with the polar head groups oriented out towards the extracellular side and the cytoplasmic side, with a hydrophobic core in between them. This is hydrophobic core. The distribution of the phospholipids is such that choline containing phospholipids are mainly in the external layer, and ethanolamine and serine containing phospholipids are in the inner layer. Okay, this one favorite topic in Viva exam. Okay, you may ask in Viva why present choline and ethanolamine. So it has various protein, carbohydrate, cholesterol scattered within it. There is what it calls mosaic pattern. This model first proposed by Singer and Nicholson in 1972. Previously, it was believed that cell membrane is a rigid structure. After this theory, we can come to know that cell membrane is a dynamic structure. Right? Fluid means constant movement of the lipid and proteins within the membrane. The components are not locked in the static position but can move laterally. Lateral means sideward or maybe forward, backward. So the cell membrane is a flexible and self-reclining structure. Mosaic means patchwork of different proteins and other molecules scattered throughout the membrane, creating a mixed and dynamic pattern. So this is called fluid mosaic theory of cell membrane. Very easy. Understand everyone? So कुछ समझ समझ जाए सब. अगर क्वेश्चन आएगा तो लिख पाओगे ना? Short note with diagram, definitely. Right? I have seen in your senior best students, some students they write just like essay. This is not a way of writing answer. You make diagram, we make flow chart, you will get better mark. And examiner like you, it is easier to read also. Okay, your answer sheet with color diagram preferably. It will carry more marks, right? Some people know lot of things, but they does not write well. Try to make a habit, because MBBS is quite long course, na? 5.5 years course, right? Next. So you understand what is fluid mosaic theory of cell membrane? Right. Next, what are the factors who influence fluid mosaic theory? Factors affecting fluidity of membrane. Please remember 4 factors. Even don't find in your test book. I make it very easy for you. There are 4 factors which responsible for fluidity of membrane. These factors are temperature, type of fatty acid tail length, and cholesterol content. These are fatty acid tail. Temperature. Next is type of fatty acids tail and tail length, and cholesterol content. These are the 4 factors which responsible for fluidity of cell membrane. We discuss one by one. Temperature. Increased temperature causes more movement of the inside the cell membrane. Okay, so increasing temperature causes increasing fluidity. Okay, more temperature means molecular movement is more, very easy, and less temperature means less molecular movement, so less fluidity. बुझते वाले? So increased fluidity, higher temperature provides more thermal energy to the lipids, causing them to move around more and spread apart. Low temperature means decreased fluidity. Lipids have less kinetic energy and pack together more closely, causing the membrane to become more rigid and less fluid. Next is fatty acid tail. Okay, in this tail we have different type of fatty acids. In your lecture class, fatty acid you will learn types of fatty acid, saturated fatty acid, unsaturated fatty acid. Okay, just remember, saturated fatty acid means there is no any double bond, and unsaturated means there is more number of double bonds. Okay, sir? So in fatty acid tails, decreased fluidity in case of saturated fatty acids like oleic acid. Okay, here no double bonds, so making the hydrocarbon chains straight. These straight chains can pack together tightly, increasing intermolecular interactions and reducing the membrane's fluidity. In case of unsaturated fatty acid, there is increased fluidity. Unsaturated fatty acid example, linoleic acid, arachidonic acid, they contain double bonds which create kinks in the hydrocarbon chains. Okay, so there is more fluidity. Okay, next is fatty acid tail length. Longer fatty acid tails need decreased fluidity because longer hydrocarbon chains have a larger surface area, allowing for more intermolecular interaction. This van der Waals force. These increased interactions cause the membrane to be less fluid. Next is shorter fatty acid chains. Here increases fluidity because shorter fatty acid chains have a smaller surface area, leading to fewer intermolecular interactions and increased fluidity. Any question? সবাই বুঝতে পারলে? ব্যাক বেঞ্চ বুঝতে পেরেছ? সমাজ কে না? হ্যাঁ, ফাইন. Next, cholesterol content. Here, just remember opposite thing. More cholesterol means less fluidity. Less cholesterol means more fluidity. Okay. In high temperature, cholesterol decreases fluidity by interacting with fatty acid tails and pulling them together, which makes the membrane more rigid. And in low temperature, the cholesterol increases fluidity by preventing the fatty acid chains from packing together too tightly and solidifying. Yes, sir. The cholesterol does do opposite things. Please remember. So 4 factors are there to regulate the fluidity of cell membrane. Please share one. Anyone volunteer? क्या बोलो? तुम बोलो. दाओ. हाँ बोलो. Yeah, yes. You. Yes. You. हाँ, yes. बोलो. What are the factors that regulate fluidity of cell membrane? बोलो एक बार हमसे एक्चुअली बोलो टेम्परेचर नेक्स्ट fatty acid tail and length of fatty acid tail. Okay, backbench, please tell. बोलो cholesterol do what thing in fluidity? Just do opposite things. Please sit down. Fine. So you can write in your exam fluidity of submembrane. This thing you have to write. Next, membrane protein. Okay. In cell membrane we have some proteins. They are emitted in the phospholipid bilayer, carrying out most of the membrane specific biological function. There are three types of proteins. One is peripheral proteins. For example, this is peripheral protein. This is cell membrane bilayer. This is peripheral protein. This is some integral protein. This is like example. This is integral protein. They can seen both outside and inside. They can emigrate to whole cell membrane. And another thing is lipid anchored proteins. These proteins are attached with lipids molecules, okay, with some force. Okay, please write down this thing. You may not find out in your textbook. Three types of proteins. Please remember, one is peripheral proteins, integral membrane proteins, and lipid anchored proteins. These proteins are temporarily associated with the membrane through weak non-covalent interactions with the lipid surface or other integral proteins. They can be easily detached with some solvent or change in pH, like alkaline media. Okay. Example, pectin. This protein present in the red blood cell surface. Next is integral membrane proteins. These proteins are permanently attached to the membrane and can be removed using detergents or other harsh solvent that disrupt the lipid bilayer. Once you have to destroy the lipid bilayer, then you can find this. So these proteins are embedded protein. You cannot find it easily. Example, transmembrane proteins. These proteins are span the entire membrane, exposing part of both the inside and outside of the cell. For example, this is transmembrane protein. You can see on outside as well as inside. They are antibiotic. Antibiotic means they have both hydrophobic and hydrophilic nature. Antibiotic. With hydrophobic region in the membrane core and hydrophilic region facing the aqueous environment, that is outside. Okay. Example, single pass. There is crosses the membrane once. One single peptide chain is there. Like the red blood cell protein glycophorin A. This is example. Single pass. And this multi-pass. Maximum G protein coupled receptors are multi-pass. You will read in future lecture classes. G proteins. Okay. Some G protein stimulatory, some inhibitory. They act as receptor for some signal like hormone signal. Okay. Right. Both factor signals like this. We have to the membrane multiple times such as G protein coupled receptors. Other thing is beta band proteins found in the outer membrane of some bacteria, mitochondria, chloroplast, these form rigid barrel-shaped pores. These are called beta barrel proteins. These are all integral membrane proteins. Next, lipid anchor proteins located entirely on the one side of the membrane. These proteins are covalently linked to a lipid molecule inserted into the bilayer. The attached lipids act as anchor. Anchor means hook-like, tethering the protein to the membrane. Example, acetylcholinesterase, this one enzyme, alkyl phosphatase, these are proteins. Okay. So you may find in the exam a short one about transmembrane protein. You have to write all the things. So okay. Again, membrane proteins are of three types: peripheral proteins, integral membrane proteins, and lipid anchor proteins. Example: peripheral proteins, pectin, present in RBC. Integral membrane proteins, there are three types: single pass, multi-pass, or beta barrel proteins. Single pass example: glycorin here. Multi-pass example: G protein coupled receptor. And beta barrel example: chloroplast. Okay. Mitochondria found in other outer membrane of some bacteria, mitochondria, and chloroplast. They form rigid barrel-shaped pores. And this is lipid anchor proteins. Example: acetylcholinesterase, alkyl phosphatase acid. Next, key function of membrane proteins. These are function of transport, act as a channel or carrier to move ions, nutrients, and other molecules across the membrane. Signal transduction function as a receptor that binds a chemical messenger relaying signal from cells exterior to the interior. Enzymatic activity catalyze various metabolic and chemical reactions within the membrane. And cell-to-cell recognition, glycoproteins serve as signifying tags that allow cells to recognize each other, which is crucial for immune receptors, immune response. Right? This is the function of cell membrane proteins. Next, specialized membrane structures like tight junctions. Probably you have some short group discussion last day. Tight junctions. তোমাদের হয়েছিল? হয়নি এখনো? Tight junction. Okay. Tight junction means when the two cells are closed in approximation in certain area, instead of 4 bilayers, 4 layers, we only have 3 layers of plasma membrane layer seal. So this is example. Here you can see 1, 2, 3, 4. Okay. In tight junctions we have literally 3 layers, 1, 2, and 3. You cannot see there. These junctions required for passing some ions like calcium ion and other small molecules to pass one cell to another via narrow hydrophilic pores. So these channels are used for some sort of communication between cell to cell. Absence of tight junctions are implicated in loss of contact inhibition in cancer cells. Okay. Tight junction also seal of subepithelial species of organs from the lumen. They contain specialized proteins such as occludin, clodins, and also adhesion molecules such as catenin, and vinculin. So example tight junction proteins. These are example: occludin, clodin, catenin, and vinculin. These are some favorite topic for MCQ exam. MCQ. In UT exams, you may find this kind of question. Next is myelin sheath. Myelin sheath present in nerve cells. Nerve cell sets axon and dendron. What is axon? What is dendron? This is axon. Dendron is attached with the cell surface, neuronal cell surface. Okay. So myelin sheath is a membrane of Schwann cells condensed and spiral many times around the central axon. So you can see this is the axon membrane, axon, and the myelin sheath is attached with the axon and with spiral shaped. Here you can see the nucleus is pushed one side with cytoplasm also, and we can see here some junctions. These are called node of Ranvier. Myelin is composed of sphingomyelin, cholesterol, and cerebrocytes. Myelin sheath thin out the certain region called nodes of Ranvier. Due to this arrangement, propagation of nerve impulse is wave-like, and speed of propagation is also increased. On stimulation, there is rapid influx of sodium and calcium, so that depolarization occurs. What is depolarization? In every cell, we have a cell is a resting state present as polarized state. That means cell inside is electrically negative. Okay, near about 90 minus 90 millivolt. This is because presence of lot of protein. Protein is actually negative in nature, and less number of sodium ion inside. Yes, sir. So when the cell gets stimulated, it depolarized. That means he lost his polarization. So as a result, what happened? Some impulse generate. You will read it thoroughly in the physiology classes. Depolarization. So in stimulation, the rapid influx of sodium and calcium occurs, so that depolarization occurs. Bondage gradient is quickly regained by ion pumps. The ion flow in and out of membrane only where membrane is free of insulation. Say example, this is the free of insulation. This area we can see membrane is free of insulation, hence the wave like propagation impulse occurs. In some disease like multiple sclerosis, you heard the term multiple sclerosis, a disease where progressive degeneration occurs of myelin sheath. Okay, so in multiple sclerosis, demyelination occurs in discrete area and the velocity of nerve impulse is reduced, leading to motor and sensory deficit. Very bad disease, multiple sclerosis. Okay. So there are some example of specialized membrane protein like tie junction, myelin sheath. Some other example like microvilli. Okay, what is microvilli? In intestinal epithelial cells, we have seen some projections outward from the cell. Okay, these are called microvilli. So microvilli in intestinal epithelium cells and pseudopodia of macrophages are produced by membrane invagination. Some projections. So in this macrophage, you can see some projections like this. Microvilli. It is equal to nature of the membrane. Microvilli. Then this membrane organelle, that is intercellular organelle, like mitochondria, endoplasmic reticulum, nucleus, lysosomes, may consider as various variant of plasma membrane. The percentage of protein content varies from 20% of myelin sheath to 70% in inner membrane of mitochondria. Okay. In my membrane organelle, there are also some proteins. Next, cytoskeleton. So, as I am standing here, why this process becomes of skeletal structure. I have bones in my body, so giving me support. So I can stand, I can walk. I have some shape. Like this, cell also has some cytoskeleton. This is responsible for shape of the cell, its motility and chromosomal movement during the cell division. The cytoskeleton is made up of network of microtubules and microfilaments. Example, spectin and ankyrin. These are also again very favorite question as a MCQ in NEET PG. Spectin and ankyrin. Tubules consist of polymers of tubulin. Here you can see this is nucleus. Here you can see some projections. These are microtubules. These are microfilaments. This is cell membrane. I am taking from Basidagon. Okay. Next, transport mechanism. I am not going to discuss the details in this system because I will read in details in physiology classes. Just remember transport mechanism. There are three types of transport mechanism: passive transport, active transport, and some pumps. Okay. Transport means to transport something through the cell membrane. This permeability of substances across the cell membrane is dependent on their solubility in lipids and not its molecular size. Water soluble compounds are generally impermeable because of hydrophobic pore and require carrier mediated transport. This factor is very important to withstand unwanted substances to go inside the cell. They protect the cell while permitting the entry of molecule necessary for cellular metabolism. There are three types of transport mechanism: passive transport, active transport, and pumps. Okay. Pumps that can drive molecules against the gradient using energy. So example, you are living in fifth floor, so it requires water. How can you get water? We require a pump to pump water from downside to upper. So it requires a lot of energy. Likewise, we have some pumps like sodium pump, calcium pumps. They work against the gradient, concentration gradient. So let's discuss one by one. Passive transport, again we have some passive transport means they just pass through easily without any concentration gradient, without any energy. So example, oxygen, carbon dioxide, these gases, this same pass through cell membrane easily by passive diffusion or passive transport. Okay, that doesn't require any energy. Even that doesn't require any transport protein also. Yes, sir. This is called simple diffusion. Next is saturated diffusion and some other is ion channels. So simple diffusion is solutes and gases enter into the cell passively. They are driven by concentration gradient. The rate of entry is proportional to the solubility of their solute in the hydrophobic pore of the membrane. Simple diffusion occurs from higher to lower concentration. This doesn't require any energy, however it is a very slow process. So require fast process, that is called facilitated diffusion. This is a carrier mediated process. Important features are the carrier mechanism could be saturated which is similar to the substrate situation of enzyme. Structurally similar solutes can contribute to inhibit the entry of the solute. Facilitated diffusion can operate bidirectionally. They can go inside, they can come outside. This mechanism doesn't require energy, but the rate of transfer is more rapid than simple diffusion process. This one is very important mechanism. They may ask in the Bilayer exam, even sign short note, ping pong mechanism. Okay, what is ping pong mechanism? So, example, this is a transport protein. It is embedded in the cell membrane. In pong state, here you can see some solutes. When they attach with this protein going conformational change, and then achieve this state. This is called pong state, and then this solute is detached from the membrane and released inside the cell. Okay, and then again these proteins again go back in its previous conformational state. This is pong state. Very easy mechanism. This is called ping pong mechanism. This is one type of facilitated diffusion. Visually, but this is a unidirectional state. Here, solute can enter, it cannot exist. Okay. So you can see the carrier molecule can exit to conformation, ping pong state. In the pong state, the active site are exposed in the exterior when the solute binds to the specific site. You can see here this pong state. This is outside, and then there is protein going conformational change. There is its shape is changed. Once the solute is attached, and then it achieves this ping state. In this state, the active site are facing interior of the cell, where the concentration of the solute is minimal. Again, this system works only when concentration gradient is less, more to less concentration area. Yes, sir. Again, once this solute is detached from this molecule, this protein, again this change reshape this previous structure, this pong state. By this mechanism, the inward flow is facilitated, but the outward flow is inhibited. Example is hormones regulate the number of carrier molecules. For example, the glucose transport across membrane is facilitated by facilitated division involving the family of glucose transporter. Okay. So by this way, glucose can enter into the cell, ping pong state. Okay. Very good. Next, ion channels. These are membrane have special devices called ion channels. Ion channels are transmembrane proteins. They allow the selective entry of ions. Features: they are transmembrane protein. They are selective transport of one particular ion. The regulation of activity by voltage gated, ligand gated, or mechanical gated process. Okay. Different channels are available for sodium ion, potassium ion, calcium ion, chloride ion, etc. Transport through the channel is very quick, very fast. Ion channels. There are some clinical indications of ion channel disease like sodium channel. This channel require when we use local anesthetic drug like cocaine or xylocaine. They block sodium channel. Okay, you will read details about in pharmacology classes. Next is Liddle disease. The sodium channel in the renal epithelium is mutated, resulting in excessive sodium absorption, water retention, and elevated blood pressure, high blood pressure. Next is potassium channel mutation. This is a long QT syndrome. This is featured in ECG. Okay, QT interval you read in lecture, future lecture classes. What is QT interval? Just now, remember long QT interval disease. This is because of potassium channel mutation. Next is lower channel disease. The role of GABA. This one is inhibitory neurotransmitter. GABA and glycine is inhibitory neurotransmitter. Is attributed to the ability to open the lower channel at the post-eruptive membrane. Okay. Next is cystic fibrosis. In this disease, one gene is here called CFTR gene, which is a fluid transporter gene. The secretion of respiratory tract and pancreas become thick. So, patient may die because of respiratory failure or pancreatic failure. Cystic fibrosis. Okay. Next is retina. The excitation of retina rod by photon is by closing cation specific channels resulting in hyperpolarization of the rod cell membrane. This light reduced hyperpolarization is the major event in visual excitation. Okay, you read in details about when you read vitamin A, vitamin lecture classes. Vitamin A. Okay. Next, active transport. The cellular feature of transport. This form of transporter equals energy. Energy comes from degradation of ATP. ATP. International type factor. About 40% of cell energy used for this process. This is the unidirectional process. It requires several integral proteins called transporter. Example, sodium, potassium, ATPase transporter protein. Air transport system is saturated at higher concentration of solutes. Okay. So you can see this is exterior, this is hydrocell. Here ATP is required, ATP degraded to ATP, and then this solutes go outside by this transporter protein. Transporter susceptible to inhibition by specific organic or inorganic compounds. Example, sodium transporter. Pump, sodium pump. This is one example of active transporter. The cell is low intracellular sodium, but concentration of potassium inside the cell is very high. Okay, this feature is required to keep the cell in polarized state. Polarized means they have two poles, positive and negative pole. Yes, sir. Cell maintain polarized state. So, to maintain polarity, cell require this transporter. Outside of the cell, sodium content is very high. What is the blood sodium level? Jana kyu? Normal sodium ion concentration in plasma. Dono na? 135 to 145 milliequivalent per liter. Sodium ion 135 to 145 milliequivalent per liter. Potassium 3.5 to 5.0 milliequivalent per liter. Okay, this is an example. Calcium ion 9 to 11 milligram per deciliter. Okay, so this form required, they are specialized protein, they have some pairs of subunits like alpha 2, beta 2 subunits. Calcium pumps, these are pumps, they have an ATP dependent calcium pump also function as regulate muscle contraction. Okay, so calcium level is very low outside, one of the calcium go inside, then muscle will contract. So a membrane system called subcutaneous reticulum is found in skeletal muscle, which regulate calcium concentration and around the cell fiber, muscle fiber. In resting muscle, the concentration of calcium around muscle fiber is low. Fast stimulation by nerve impulse resulting in sudden release of large amount of calcium ion. This would trigger muscle contraction. The function of the calcium pump is to remove cytosolic calcium and maintain low cytosolic concentration so that muscle can receive the next signal. For each ATP hydrolysis, 2 calcium ions are transported. Okay, these are some features of pumps: sodium pump, calcium pumps, etc. And transport of large molecule, we have some secondary vesicles, exocytosis, endocytosis, pinocytosis, receptor mediated endocytosis, phagocytosis are there. Yes, so actually we are running out of time. We have only 1 year to complete your syllabus. So this is not possible to cover everything in your lecture classes. Okay, you have to go through textbook and read carefully. Okay, we can tell you what the summary or we can suggest you what the thing you have to read. বুঝতে পারলে? তো এগুলো পড়বে কি তোমরা? Any question? সবাই বুঝতে পেরেছো? Very easy. I have to read this thing. I do not go into details. Endocytosis, pinocytosis. What is endocytosis? We engulf something inside the cell. What is phagocytosis? We engulf some microbe like bacteria. Okay? They engulf bacteria from outside, they come inside the cell and then lysosome attack this bacteria, they kill bacteria. What is pinocytosis? You can say the drinking of fluid by cell. This is a pinocytosis. This is like endocytosis. Okay?